And today, the focus of this presentation will be really I have a strong focus on below ground chemical interactions. And I built this presentation to kind of brush over the main mechanism that could be of relevance for developing intercropping systems. So I have two main goals in this lecture. The first one will be to kind of shed some light on the importance of chemical interactions for intercropping. And I will do this using various examples that I found quite exciting from the scientific literature. I will use some bits of my research as well, but I didn't want to focus only on my research. Also, because I'm mostly working on grasslands, I also wanted to showcase some examples that I found particularly exciting from the recent literature that has been published in the last years. And I hope you will find it interesting as well. But first, another disclaimer a bit about my background and what I'm doing. So I actually have an agricultural background. I did my PhD on studying plant-plant interactions in cropping systems, looking specifically at below-ground chemical interactions. And then I transitioned a bit during my postdoc to more plant community ecology topic, so more in natural systems. And now I'm actually working as an assistant professor in Utrecht, mostly focusing on grasslands and forest ecosystems, actually. So I left a bit the agricultural sector, but there is still quite a lot of relevance when you study below-ground chemical interactions in natural systems. There is a lot of relevance to this research for more diversified systems in agriculture and cropping systems, especially. So this was for my disclaimer. So I saw along the walls here, there is quite a lot of diversity of researchers in the room. You're all working on, of course, on intercropping or strip cropping, but from very different perspective. And I was curious, who is doing some work related to chemical interactions in cropping systems? Show of hands. No, a little bit. I mean, chemistry is everywhere, so I guess you can always say that. Good. That means that maybe a lot of the things I will talk about will be new to you. So as a researcher, I work quite a lot in this field, the field of chemical ecology, which is similar also to what Lilong is working on. And it's a discipline of ecology that basically studies interactions between, in my case, plant individuals of the same or different species, and these interactions being mediated by chemical compounds that are produced by plants or other organisms as well. And what I often tell my students is that when you look at plants, and if they had like some kind of master degree, they would probably be chemical engineers, really, because they are extremely good at producing specialized metabolites that have various ecological functions. So I just listed a couple of chemical compounds that you're probably all familiar with without knowing it. So, for example, when you mow the lawn in your garden, you quickly have this fresh green smell coming to your nose. This is because plants, when they are damaged, they quickly release this green leaf volatiles into the atmosphere, and you smell them. And these have also important ecological functions, and I will go back to this a bit later. You're all familiar probably with essential oils that we extract from flowers to make perfumes, for example. And below ground, plant roots are also very good at producing chemicals and releasing it into the rhizosphere. And these chemicals also mediate a lot of interactions below ground. And this is something that I would like to discuss a bit more in this presentation. But let's start from the beginning. And I wanted to showcase here some examples of why chemical compounds are actually important when you study interactions between living organisms. And I just wanted to showcase some examples. For example, flowers, we know that they are very good at producing volatiles that can attract, for example, pollinators. And this is a very important ecosystem function also in cropping systems. But what you maybe not know as well is that when plants are, for example, attacked by a herbivore or pathogens, they can also answer to this attack by releasing chemical compounds into the atmosphere, also in the form of volatile compounds. These volatiles can then diffuse into the air and be perceived by other organisms. It can be another plant, but in this case, it would be a natural predator of the herbivore that is attacking the plant, which allow this parasitoid in this case, which is also showcased here, to basically reach the site where the herbivore is attacking the plant and basically will then attack the herbivore. But all these interactions that are mediated by chemical compounds above ground, you can also find similar types of interactions below ground. And I will showcase some interesting examples of mostly plant-plant interactions below ground. But I just want also to highlight the fact that those chemical interactions are not only limited to plant-plant interactions. I mean, if you look at the soil is an incredibly diverse ecosystem. You have plants growing in them, of course, but I saw that some of you are working with nematodes, for example. You also have a lot of bacteria and fungi and viruses. And all these organisms are interacting with each other and, of course, will affect plant performance. And this is of relevance because of, I don't know if you saw this paper, I found it quite interesting because it shows that a lot of, like more than or close to 60% of the species that we know of on Earth are actually tied to the soil environment. So the interaction that happen below our feet actually carry a lot of weight and have a lot of ecological relevance. Also, if you think about cropping systems. I would like to show you a typical example that I learned about when I was actually a PhD student a couple of years ago. This is, for me, a textbook example of below-ground chemical interaction between plants and other soil organisms. So in this case, you have a maize plant that grows into the soil, and maize, can be attacked below ground by different types of herbivores. And in this situation, this is the larvae of a coleoptera that is called the western corn rootworm. And these larvae feed basically on the roots of maize, which is, of course, very damaging for maize itself. And so as a way to defend itself, some maize varieties are able to release a very simple chemical signal into the soil, which is a volatile compound that we call E-beta-caryophyllene. You can just forget the name, but this is basically a smell that the roots of maize are releasing into the soil when they are under attack by the specific larvae. And researchers in Switzerland, at the University of Neuchâtel, in the team of Ted Turlings, they were interested in finding out why maize is doing this, basically. Why is it taking so much energy to produce this chemical? And they found out that actually this chemical compound can be perceived by all these microscopic worms, these nematodes that you see on this picture, and it will attract the nematodes to the site of attack. And these nematodes have a characteristic because they are entomopathogenic. That means that they are able to actually attack the larvae that is attacking the maize plant. So to defend itself, the maize is actually releasing a chemical signal into the soil that attract entomopathogenic nematodes that will ultimately kill the larvae that is damaging the maize. And I found this example to be quite fascinating because it really shows the importance of below-ground chemical interactions also in this context in cropping systems. But if you look at plant-plant interactions now, so really how plants of the same or different species interact with each other, they can do this in many different ways as well. So you are certainly familiar with the perception of red and far red ratio and so on. But above ground, plants can also release volatile signals, for example, when they are attacked by herbivores or when they are attacked by pathogens. And these volatile signals can also be perceived by a neighbor, a plant neighbor, either the same or different species. And this will trigger defense mechanism in neighboring plants that will allow them, that will basically prime the plants and will be able to respond faster and actually stronger when they will be under attack themselves. What was always of high interest to me basically is not necessarily what happened above ground. This is quite well documented, but this is what happens below ground, because I mentioned that plant roots are very good at also producing chemical compounds. Longley talked about this in his lecture. Those root exudates, they also play very important ecological roles. And so if you look at all these chemical compounds, this diversity of chemicals that you can find in the soil, it's kind of a mess, really, what's happening in the soil. That's why it's quite challenging to work in this type of system. But if you imagine you extract the water that you find in a soil sample, for example, and then you look at all the metabolites, all the chemical compounds that are present in this water sample, they come from very different places, from very different origins. Of course, some will be produced by plant roots. So this is just one process, root exudation. And I will come back to this process a little bit later in the next slides. But when you think of it, chemicals that can also be taken up, but also released by microbes that you can find in the soil in association with plants or other free living. Plants also produce a lot of organic matter, a lot of litter that at some point will also get mineralized, and this mineralization also release and produce quite a lot of chemical compounds that can also affect plant performance. And those chemicals can also interact with the soil matrix, being immobilized or being transported in different other parts of the soil. So these chemical interactions that take place in the soil are basically very dynamic, right, in both space and time, which makes them quite challenging to study. So if we look now for a moment to one of these processes a bit more in detail, if you look at root exudation per se, is anyone here doing root exudation measurements in their research? No? Yeah? Okay. Yeah, a little bit?
I plan to.
You plan to. Right, that's great.
I mean, too.
You too?
I'm planning. Yeah.
Yeah, it's always there is a long planning phase to do that. I totally get that. So for those who are not familiar, so I'm not familiar with root exudates. I recommend this very nice paper from Eva Oberger and David Jones, because they really provide a good overview of what are basically how we define root exudates, how different they are from what we call rhizome deposition, and basically they also provide some tips on how you can collect them and analyze them. So this is a good starting point if you want to know more about that. But they define it as the soluble organic compounds that have either a low or high molecular weight that are basically released by plant roots into the soil. And those root exudates, this figure basically summarizes some of the ecological functions of these root exudates. And I would mostly focus on what's happening in the center over here. So basically what happens when you have a plant, here in this situation is probably a maize plant, that release these exudates into the soil. They can be involved in signaling processes. So that means plant detection of neighbors between plants that grow next to each other. But they can also have indirect effect on plant neighbors via acting or on soil microbial communities and changing, for example, the structure or the composition of that community. So in this figure, I took it from a nice review paper from Wang et al. This figure attempts to basically summarize the main types, the main roles of root exudates in plant-plant interactions, in plant-plant signaling. So on one side, on the left side here, this figure illustrates that those chemicals can mediate intraspecific interactions. So basically interactions between individuals of the same species. And they are, for example, involved in kin recognition. So parents being able to detect their own kin. There is some evidence for that. But those exudates seem to also play a very important role in interspecific interactions. That means mediating interactions between different individuals of different species, which would lead to, for example, a neighbor to detect the presence of another plant and avoid it and be able to place its roots in another location of the soil. So this type of interaction can affect, for example, how plants distribute their roots in the soil, which is something that Long nicely mentioned in his lecture. But if I want like to show you, showcase some examples, research examples of that with actually relevance for intercropping systems. So this is an example of research that has been done with wheat and quite a number of other plant species. So basically, they have an experimental setup like this, where they grow wheat plant in the center, and they surround it with individuals of another plant species. And they tested this type of interaction with over 100 different plant species in this system. So it was quite a substantive study. And what I liked about this study is that they actually teased apart or looked at two different processes mediated by below-ground chemicals. On the one hand, they basically look at how the wheat is able to basically perceive the presence of another species growing in its vicinity. So this is called neighbor recognition, and they identify some signaling chemicals, like for example, jasmonic acid and loliolide, that were produced quite generally by all the species they tested in experiments. So there's a very general chemical compound that seem to be produced by a lot of different species in their exudates. And these chemicals can then be perceived by the wheat plant growing in the center. And as a reaction to that, the wheat then start increasing the production of this specific chemical called dimboa. It's the names of these chemicals are always a bit complicated, but this is a basoxynoid that has allelopathic properties. Have you heard about this word allelopathy before? Do you know what this is? Yes?
So those are basically kind of natural herbicides that plants produce to damage their competitors.
Yeah, it's actually, if you look at the original definition of allelopathy, what you describe is actually a subpart of it. That means it's true that for a lot of people, allelopathy means, for example, you have a plant that produces a chemical, and this chemical can then be basically have an effect on individuals of another species, and usually a negative effect. That means it will either inhibit its germination or lower its growth. And in this context, this is basically what the dimboa would do. So that means the wheat is able to produce this compound, which would then have a negative effect on the growth of the other species growing with the wheat. And they noticed that this effect was actually dose dependent. So that means the more neighbors you have, the more of this species, the more of this neighbor you have growing along the wheat, the more the wheat starts to produce this dimboa compound. So you have like a two-step process where first the neighbor is detected by the wheat, and those chemicals are the ones seem to be the ones acting in this interaction. And then as a response, the wheat produces this allelochemical that will then affect the growth of the neighbor. But if I go back to this definition of allelopathy I mentioned, it was much broader at the beginning. I think it was in 1996, the International Society for Allelopathic Research defined actually allelopathy in a very much broader sense. Like it was basically any process that involves the production of secondary metabolites by, it could be a plant, could be a microbe, could be or another species that will basically affect another individual, but either negatively or positively. So that means positive effect were also included in the original definition of allelopathy. But in people's mind nowadays, it's really mainly focusing on a negative effect of one plant to the other, mediated by chemical compounds here. This example actually showcases a direct effect of chemicals. That means you have a plant A that produces a chemical that is being perceived by plant B, and this chemical has a direct effect on the plant by, for example, inhibiting its growth. But these chemical interactions can also be more indirect and mediated by the soil microbiota, which is something maybe you are more familiar with. And in this paper, they also work with an intercropping system. So they actually were growing maize in association with, I think it was faba bean in this example, although I would have to check. But what they found is that they basically compared maize growing in monoculture, so just maize alone, with maize growing with faba bean. And then they looked basically at the rhizosphere metabolites that were present, so the chemical compounds that were present in the rhizosphere of maize. And what they found is that in an intercropping setting, the rhizosphere metabolites around maize roots were actually a bit different than when growing in monoculture. And some of these chemicals, like you have shown here on this graph, were actually more abundant when maize was growing in an intercropping setting. And when they pushed their investigation a little bit further, the authors of this paper found that actually these chemicals didn't have a direct effect on maize itself, but actually had modulated the assembly of the rhizosphere microbiome. So that means the plants produce chemicals that affected, for example, the structure, but also the diversity and the complexity of the microbial communities present around maize roots. And this change in microbial communities due to the production of these chemical compounds had then a positive effect on the nitrogen and phosphorus uptake of maize itself. So that means that by growing in an intercropping setting, those underlying chemical interactions led to an increased growth of maize in the sense with a higher, for example, nitrogen and phosphorus content, but also higher biomass production both above and below ground. And so this is more of an example of an indirect effect of these chemical interactions. And in his lecture, Longley showed you also the effect of those chemical interactions on nodulation, on the production of nodules by legumes. And I would like to show you another example of that. It's an example from the team of Yan Chen at in China. They work in another intercrop system. They work on peanut as a legume species. And they had basically 3 different modalities in their field setup. They had either peanut grown alone, so in a monoculture, peanut with peanut, that's it. And they also had 2 other situations. One situation where peanut was rotated with oilseed rape, and a first situation where peanut was grown together with maize, and then followed by a rotation with oilseed rape. And they looked at how these different treatments affect the nodulation intensity of peanut as a legume. And they found very strong differences between the treatments. And actually, they found that you have a much higher nodule density in situation where you have either a rotation of peanut with oilseed rape, but the greatest nodulation intensity was found when the peanut was actually intercrop with the maize. And they were wondering, okay, they actually investigated the ecological mechanism behind this, and they found that the peanut plants, when they were grown in intercrop with maize, they were basically changing the metabolites that were present in the rhizosphere. And this change in metabolite and chemicals that composition led also to changes in the activity and growth of microbial communities present in association with the roots. And they looked separately at what happened to free-living organisms that were able to fix actually nitrogen as well. And they also look at what happens to organisms that form symbiotic relationship with plant roots. And in both cases, they found out that this change in rhizosphere metabolite led to an increased activity of free-living organisms that are able to fix nitrogen, and it also led to an increase in symbiosis between the roots of peanut and the bradyrhizodium in this case, which is a microorganism that developed these nodules with peanut roots. And so this also highlights the importance of these chemical interactions and root exudates in basically affecting nodulation and nutrient uptake in intercrop system. Now I would like to shift gear a little bit and talk a little bit more about something that I'm a bit obsessing about, which is basically more related to my research, because I'm very much interested in plants of feedback, also in agricultural context, but also natural system. Have you heard about this concept of plants of feedback? Some of you did? Great. It's actually very important in agriculture as well. This is actually why we do rotations in agriculture. But when we talk about plants of feedback, we talk about the legacies that growing plants leave behind in the soil and how these legacies can then affect the next generation of plants. And so far, when you look at the plants of feedback literature, the focus has been very much on microbes. I think some of you are studying basically or mentioned earlier that it's very bad practice to grow, for example, maize after maize after maize after maize, because you have a buildup of pathogens, for example, in the soil that actually reduce the performance of the crop over time. This is a typical example of a negative plants of feedback. And if you want to test these plants of feedback experimentally, the way to do it is usually to have like an experiment consisting of two different phases, what we call a conditioning and a feedback phase. During the conditioning phase, you would grow individual species, in this case, species A and species B, in the soil, and you will let the plants condition the soil for a while. By conditioning the soil, I mean you let the plants change, for example, the composition of soil microbial communities, you let the plants take up nutrients, you let the plants exude chemical compounds in the soil. And after a while, you're gonna take the plants out of the soil. You have a soil conditioned by species A here and a soil conditioned by species B. And then in a follow-up experiment, you would grow species A and B alone, but in soil conditioned by species A, also conditioned by species B. And you do that for all the species included in your experiment. And if you find that a species grows better in a soil that was actually conditioned by another individual of its own species, this is called a positive plants of feedback. If it's the opposite, and if the species grows actually worse in a situation where the soil was conditioned by individuals of its own species, you have then a negative feedback, which is what I mentioned earlier, and is very common also in more cropping systems. But so far, the focus has been very much on microbial mediated plants of feedback. So basically, plants change the microbiome, accumulate pathogens, and this can have follow-up effect on the next crop. But I have a slightly different view. I try actually in my research to have a slightly different view on plants of feedback. This is like more I would say the dominant view that we have in the plants of feedback field, where a plant grows in a soil, condition that soil by changing the composition of mutualis, decomposers of pathogens that are present in that soil. And these changes can then feed back to the next generation of plants. That's the idea. The way I'm conceptualizing and thinking of plants of feedback is a bit different, because I distinguish between microbial legacy, so how plants affect the soil microbiota, but I also include this how plants change the chemical legacies in the soil. That means when plants grow in the soil, as I said, they would produce root exudates, but there are also other processes at play, for example, litter decomposition, that can release, for example, self-DNA fragments that have also been shown to actually contribute to this negative plants of feedback. It was a nice paper. I didn't put it in the presentation, but they also play a role. And I was wondering how these chemical legacies, if they can actually play a role also in mediating these plants of feedback in natural systems. And so to test this, when I was a postdoc in Germany, I did an experiment involving two different grassland species. And what I did is that I grew two different types of plant communities. That means multi-species communities. I had communities consisting only of 4 species, so herb species on one side, and I had another plant community that was only consisting of grasses, grass species on the other. So 2 very different species composition. And I let those plant communities condition the soil for a couple of weeks. So I let them grow, I let them do their thing and change the biotic and abiotic properties of the soil. And then after a while, with some students, we started collecting the soil solution, so the water present in the soil from these different plant communities. And we did this for a couple of weeks, and we stored then the soil solution. And after a while, we also have to mention that we also had a control situation in that experiment where we had actually pots with just soil, no plants growing in them. It was kind of a control situation. And then as a follow-up, we took the soil solution that we collected from these different pots with different plant communities, and we applied this soil solution that contained all these metabolites that we find in the soil. And we applied it to two different species that were common in dry grasslands, Dianthus deltoides, a fauve on the left, and Festuca rubra, a grass on the right. And what we found, it was very interesting, because we found that if we applied a soil solution from a fauve community, or soil solution from a grass community to this species, nothing happened. It didn't really care about it. It was not different from the control. But when we expose this other species, Dianthus deltoides, to either the soil solution from a fauve community or the soil solution from a grass community, we noticed that it actually changed it basically had a lower root productivity. The production of roots for that plant started to go down. But what was particularly interesting, I found, is that the mechanism behind this reduction in root production was different depending on the type of chemical legacy that we applied to the plant. So if the soil solution came from a fauve community, this reduction in root foraging was mostly driven by a reduction in root dry weight. So not the root traits were not different than from the control situation. But if you apply to this specific species the soil solution from a grass community, however, the reduction in soil foraging was not due to a change in root productivity, but was due to a change in root traits. Actually, the plant started to change, for example, the specific root length, so that has negative effect on specific root length, which means that plants started to produce basically thinner roots. And this led to an overall reduction in root foraging. What I found also interesting in there, and this is something that I was kind of expected but didn't find, is that this change in root foraging didn't really translate in a difference in nitrogen uptake by the plant. So that means if you look at the total nitrogen content in plant root, you didn't find any difference there. And so this kind of set in motion kind of a research line in my career that shows where I'm trying to link and show and study how these soil chemical legacies play a role in plants of feedback, in mediating these plants of feedback, to add this additional lens in addition to the usual focus on soil microbial legacies. And recently, I was really amazed by this study that was conducted by some colleagues in Switzerland, where they had a similar type of questions. They studied plants of feedback mediated by chemical compounds, but more in a cropping setup. And they did this experiment in the field. And I found the results were quite astonishing. So they had a very simple setup. In the field, they grow two different types of maize. And these are actually colleagues from the University of Bern in Switzerland. They had a maize called the wild type. And this variety of maize was able to release a specific class of chemical compounds via root exudation. And these chemicals are called benzodaxinoid. Don't have to remember the name, but just remember that this wild type basically is able to release a specific type of root exudates into the soil. And they grow in another side of the field, a mutant of this wild type that was basically not able to release these chemical compounds via their root system. So there's very simple treatment. And they let these two different maize lines grow into the field for a couple of weeks, basically for a whole growing season. And after a while, they harvested the maize, and then they put wheat on the plots that were conditioned by these two different species. And what they found was quite interesting, because they found that the wheat responded to this treatment. That means whether or not the wheat grew on soil that was conditioned by plants that are able to release benzodaxinoid or not, it had, for example, an effect on yield. They found, for example, that the yield of wheat plants was higher when it was growing on soil that was conditioned by plants who were able to release these benzoxynoids. And the increase actually quite subtle because it's like, I think it was a 4% increase in yield. It doesn't sound like much, but if you look at actually the surface area where you grow this wheat and so on, it actually makes quite a substantial difference in amount of grains. And they also looked at, for example, whether this different soil conditioning affected grain quality and baking properties of the wheat, and it didn't. So it really increased the production of the yield of wheat, but didn't change the properties of the grain. So it kind of highlights and shows that this below ground interaction taking place in the field can also have consequences and implications for the next generation, the next crop, basically, with positive effect on yield in this situation, which I found really, really interesting. And so far, I've been mostly, and this is basically the last example I will show you today. So far, I've been distinguishing what's happened above ground from what's happened below ground. But the truth is, those chemical interactions, they are linked, right? So that what happens above ground, below ground is kind of coordinated to each other. And recently, I read this paper from the team of Ling Fei Hu, also in China. Maybe you know Ling Fei. And I was really amazed by this paper. It's really a fantastic paper, and I really, if you're interested in this topic, I would recommend you to read it, because they looked at basically what they did is the following. So they grew maize plants, and at some point they basically had added some insect larvae that started feeding on the leaves of maize. And as a response to that, the plant started to release volatile compounds into the atmosphere, which are represented in this figure by these chemicals over here. Those chemicals were then perceived by a plant neighbor that was not yet under attack by the pathogen, by the herbivore. And this exposition to these green leaf volatiles trigger some jasmonic dependent changes in the receiver plant that basically led the exposed plant to change the composition of the soil microbiota that was associated with its roots. And actually, there was a greater proportion, for example, after exposition, there is an increase in beneficial soil bacteria that are associated with those plant roots. And this is just triggered by this exposition to these volatile compounds here. There was no attack by herbivore whatsoever. And once they removed the plant from the soil and look at basically how these soil legacies carried out to the next generation of plant, basically they took the soil from this exposed plant here and they grew then another maize individual on that conditioned soil. And they found that this individual actually grew better. It grew better, and it actually was more defended against, for example, an attack by a herbivore as well. So this shows that above these chemical interactions happening above ground, but also below ground, can really mediate these plant soil interactions and actually help crops grow better and being better defended in the face of pests like insect herbivores in this situation. And I found it very interesting, and I'm kind of thinking now along with these results and looking at whether there are implications, for example, for ecological restoration and so on. So to just the main, very simple take-home message from this presentation by showing you these examples and this from the literature and a bit from my research as well. I hope I kind of convince you that this that trying to better understand these chemical interactions actually matter, not only for just the sake of ecology, but also that it can have implications for a cropping system because it could also help us in fine to create more diversified cropping system, but also a cropping system that are more sustainable and eventually more resilient system. So this is it from my side. I'm trying to be as short as possible, and I'm happy to take questions if you have any.